Hello students, this is Dr. Faizan Mirza. I'm covering the topic of mutations and the type of mutations in this content of my current video. So type of mutation, what exactly is a mutation? Mutation could either be a chromosomal mutation or a genetic mutation. What I'm covering here is genetic mutation. Genetic mutation basically occurs as a point mutation. What do you mean by point mutation? Point mutation means a change in a single nucleotide at a specific point. The point mutation could occur either by substitution, deletion, or insertion. We'll be discussing all the three in detail. Let's discuss substitution first. Now, as we know that a codon is a triplet of bases and one codon actually refers to one amino acid, or it's basically a code for a single amino acid. So if I'm having the first codon here, the second codon here, the third codon here, the fourth codon here, I know that these four codons will code for these four amino acids in the very sequence. The first codon, the second codon, the third, and the fourth. And the sequence in which the amino acids are joined together, the order in which they are joined together, that's the primary structure of a protein. Inside a codon or in a codon, since it's a triplet of bases, there is a first position, the second position, and the third position. So the first position of the first codon is C, the second position of the first codon is A, the third position of the first codon is occupied by A. So this is the first codon. Come to the second codon. In second codon, the first position is occupied by the nucleotide T, the second by T, and the third by T again. So for every codon, there are three positions: the first position, the second position, the third position. Together, this triplet will make up a code, and this code will be telling you which amino acid will be placed there. Now, what happens in substitution? In this case, I have taken an example from the book in which the TTT codon turns into TAT. It's a point mutation. Point mutation means change of a single nucleotide, in which T got substituted by A. Which position changed? The second position got changed. So in this case, you can see that the second base of the second codon, T, is substituted by A. In this case, the amino acid got changed. And because the amino acid got changed, it means that this mutation will result in an altered sequence of the polypeptide. The primary sequence will change, which obviously will lead to the changes in the secondary and the tertiary. And probably, if the quaternary structure exists, then they will be changing as well, because we know that the folding depends on the placement of the amino acid within the polypeptide chain. So this amino acid might just be having a different R group. It might be polar, it might be non-polar, and the folding can change, and the function of the protein can be changed as well. So coming back, if it's a substitution mutation, in this case, we know T got substituted by A, and since the change of codon from triple T to TAT in this case changed the amino acid as well from lysine to isoleucine, if the amino acid is changed because of the change in codon, we say that's the missense mutation. Let's take another example. Now again, you have four codons, and in these four codons, again, they are coding for these four amino acids in the very same sequence. You can see the first position is valine, the second is lysine, the third is leucine, and the fourth is glycine. They will be provided to you. Now, let's suppose if in this category, if we consider that the third base of the second codon, the third base of the second codon means this is the second codon, and the third base is C. Let's suppose if the third base C gets substituted, and this time it gets substituted by C still. If it's still getting, like, if it's C, or let's suppose, since this initially was not C, this initially was a T here because it was a triple T, and triple T codon was coding for the amino acid lysine here. If this double T turns into double T C, so instead of triple T, it's TTC, the third position of the second codon got changed and got substituted by C. Initially there was a T at this position. Now still we know that double TC and triple T, they both code for the same amino acid. You can use the tables which are provided to you in the questions, and these are provided in the appendix of the books as well. If you look for the codon double TC and you look for the codon triple T, they both code for the same amino acid lysine. So a mutation that changes triple T, the codon triple T, into a codon double TC, the mutation is very much there. The point mutation occurred, substitution mutation occurred as well, but this change of codon did not result in any change of the amino acid, so this is called as a silent mutation. So silent mutation and missense mutation are both basically substitution mutation, in which in the former one, the change of a codon changes the amino acid. In the latter, the change of the codon does not change the amino acid. How do you define substitution mutation? Basically, substitution mutation is simply a base getting substituted in place of a base that was already there at that position. So base gets substituted at a specific point or at a specific position within the codon. and the altered codon can then code for a same amino acid or the different amino acid, as already covered. Apart from this, which kind of mutations can occur? We can have a deletion mutation or an insertion mutation. What's a deletion insertion mutation? If deletion is occurring or insertion is occurring, they both allow frame shift mutations. Now what's a frame shift mutation? A frame shift mutation changes the reading frame of the mRNA altogether. For example, the first codon was CAA, then TT, and then GAA, and then CCC. This was the actual sequence of the bases present in the DNA. Now let's suppose if a deletion or insertion will occur. Deletion means you subtract a base from the existing sequence. Insertion means you add a base in the existing sequence. We know that every codon can have 3 bases, not more than 3. So if deletion occurs, that means one of the base will get subtracted. If insertion occurs, one of the base will get added. It will change the sequence in which these codons are read, and that's called frame shift. For example, if the CAA and then double T, triple T here got added, or you can say this got deleted. This here, it gets at this position, the third position, we have an insertion of A at this side. So this codon triple T turns into double T A. So this additional A, when it comes here, so this codon cannot be triple T anymore. The codon becomes double T A. So again, this is a point mutation. But this codon cannot have more than 3 bases. So initially it had triple T, but now it is having double T with an A. So what about the T which was already there at the third position in this codon? This T will go and will be read in the next codon. So the next codon was initially GAA. Since this T gets placed here, so this codon becomes TGA. And when this becomes TGA, this initially was GAA. The third position again of this codon will go into the next. So this will become ACC from triple C, and this C will again go in the next codon. So what has happened? We know that with insertion there is an additional base there in the existing sequence. So frame shift occurs. Frame shift means that the reading frame changes altogether in the region where the insertion occurred, and the region which was preceded by that insertion, that stays unaffected. The same goes for deletion. Now in deletion, we know, as I've already mentioned, that a base from the existing sequence will be deleted. So in this case, let's suppose this triple T codon, I'll take the example of the same triple T again. If this triple T codon, this T, the base T, the third position gets deleted. If that gets deleted, we have G from the next codon coming and replacing it. So this G will come here, it will replace the T which got deleted altogether. So the codon triple T becomes TTG. Now with TTG forming here, it's a new codon here. What happens to the next codon, which now is AA? It will borrow C from here. So this codon will become AAC. The last codon in this case will be left with double C. So it will borrow the base from the next. And so again, we know that again, because of this deletion mutation, the reading frame changed. So this is called frame shift mutation for deletion as well as for insertion as well. And the codon which was preceded or which was before the point where the deletion or insertion occurred, this codon stays unaffected. In both the cases you can see CAA, they both are unaffected, so their amino acid will be unaffected. But whereas the amino acid which are placed ahead will be quite a bit changed because of all the reading frame getting changed altogether. That's it. Thank you so much.